Hi everybody. So we are on the last part of topic 16 and you'll be glad to know that this last part is really short. And so I'm going to do my best to summarize quickly. I want you to make sure that you understand the topics that we've discussed before this. And so come with any questions that you have during our chat session. If you recall, the last video was basically about what do you do when two different data sets disagree, or how do you even figure out that they disagree? And so we talked about the Templeton topology test, which allows you to determine whether maybe one of your data sets can still achieve the phylogeny of another data set, so that the two data sets are essentially not in conflict. So make sure that you review that and you understand it. You will have to answer a question about that on the next quiz. And then what I'm going to do now is take that last topic and talk about what happens when you have two data sets that from the outset look like they just don't agree whatsoever. And the question then becomes, could you perhaps combine the data of those two partitions that you are looking at and get a better tree? And so we're going to talk about one last test that I will introduce to you as part 3. So follow along. If you have any questions, please bring them to our chat session. The first thing we're going to do is talk about a generic data set, and then I'll come back to the carnivores because that was our model data set that we were looking at. I want you to imagine that you've got two different partitions, and here we're looking again at DNA. And imagine that you have run a parsimony analysis, so a cladistics analysis, and you've retrieved these two trees from the analysis. They're equally parsimonious and they have 34 steps each, right? That suggests that they each have 34 individual evolutionary changes on those trees. So the two partitions are equally parsimonious, but if you look closely at the relationships, they are quite different. And so you can quickly look at the trees and you can compare across, and you will notice that they are quite significantly different from one another. In one, you've got B here at the base, more close to the base of the tree, whereas in the other one, you've got species G. And so just that difference, as along with these other clades down here, suggests that these two different genes, say, which are different partitions, are giving us two very different answers. And so what do we do about that? There is a test called the Incongruence length difference test. Sometimes it's also called the Partition homogeneity test. And what this does is it determines essentially whether the two partitions that you are looking at could potentially give you a better answer or a shorter tree if you're using parsimony if you combine the two data sets. And so that's what we're going to look at. We're going to assume that we're just talking about this in a parsimony framework, but you can use it for maximum likelihood. You just can't use it for Bayesian analysis. All right, so here are the two data sets and the two partitions. And remember, a partition is basically the same set of taxa that you are analyzing a little bit differently. So one might be morphology, one might be molecules, okay? And so what we're going to imagine is that we have these two generic partitions. They are DNA, but you can have morphology in DNA as well. And what this test does is helps you determine whether the two partitions or the data sets that are being used are congruent. Congruent meaning that they say the same thing. They agree with one another. All right. And what you're going to be looking for is if the test shows that there is congruence, it means that the two partitions or the two data sets have very little conflict and that they can be combined into a single analysis. And if you find that the two data sets are not congruent, which I will show you how to interpret, is that the two data sets are in conflict, significant conflict, and thus should not be combined. Now, you still run into the issue of if you do this test and you find that the two data sets are really in conflict and they shouldn't be combined, and you've done the Templeton test that also shows you that the two data sets are very much significantly different, then you are basically going to have to explain in the paper that you're publishing that these two genes are telling you two very different stories. And at the end of the day, all that might mean is you should probably look at more genes, and that might give you a more complete story. All right, so we're going to look for whether our data sets that we are examining should be combined to perhaps give us a better answer, or whether they should be kept separate. So we are going to imagine that we're working with this generic data set, and the first step in an ILD test is that you calculate the length of the most parsimonious tree for each partition separately. So the ILD test starts with you run your analyses of your two partitions separately. They are not combined. We tend not to combine right off the bat. So partition 1 is, say, some kind of gene that you are interested in studying or that you somehow sequenced. So this is gene 1, and then partition 2 is going to be gene 2. So you run them separately, and what you get for the first tree, which we're going to call L1, tree 1, its length, L1, is 34 steps. And in addition, we also know, based on what I just told you earlier, that the length of the second tree for the second gene is also 34. They are equally parsimonious, but the trees are different in their topology. Their relationships are not the same. So what you do first is you sum up the lengths of the two partitions when they were run separately. And what would that give us? That would give us a value of L1 plus L2 equals 68. So you sum up your lengths of the two partitions that were examined separately, and that gives you your L1 plus your L2. All right, so 34 plus 34 gives us 68. All right. What we're going to do next in the ILD test, and the program does this for you, is it creates a random partition. And what you will notice on this slide is that you've got at the very top a partition assignment. So what the program does is it takes your data and it creates a random partition. This is step 2. It randomly reassigns your characters from the two partitions separately into one combined partition. So every partition is going to be sampled randomly. Here, this first character in this partition, this new pseudo partition, comes from partition 1. The second one randomly comes from partition 2, and so on and so forth. So what the program is doing is it's creating a random partition that is based on the two existing partitions. And so it randomly samples characters and builds a combined partition of the number of characters that combines those two partitions. So let's say that there are 35 characters in partition 1 and 35 in partition 2. What the random partition is it's a sum of those two partitions' number of characters. Now these are randomly selected characters, right? What you do here in the analysis is you do this a number of times. So you create somewhere between 100 to 1,000 random partitions where characters are randomly selected. It sounds kind of similar to the bootstrap, where you're creating random data sets, right? So you do this 100 to 1,000 times, and that is going to give you some range of data. What then happens with this random partition assignment is that you run the analysis and you get a set of trees. So step 3, which I've outlined on the slide, you take each partition or the combined partition, and then you look at how long the tree is for that combined partition relative to the other separate partitions that were used. So each partition by itself has a length, which we have noted were 34 each. You sum the lengths of the two partitions, which was, what was the number? 68. And then what you do for the ILD test is you do the same for your combined partition, right? How long is the tree for your combined partition? And what it does in the calculation is it looks for the partition that is built on data set 1. So that's all the characters down here that have a 1. That is going to give us a tree for that partition 1. And then you also look for the trees, or the tree that belongs to partition 2 in this combined analysis. So it's going to sound a little bit confusing, but essentially what the program is doing is it's taking your two partitions and making two pseudo-replicate partitions to see whether, when you combine randomly the characters, if you get a better tree. And so I'm going to read a little bit from the book. Those of you who have the book, and I know not many of you do, this will help reading through it. So I'm going to read through it just to clarify my language a little bit. All right. So to do a partition homogeneity test or an ILD test, we first determine the length of the most parsimonious trees for each partition. We've done that already. And then you sum up the length of those two partitions, which gave us 68, okay? Next, we randomly reassign characters to those two partitions, which is what we're looking at here. So you've got a partition that is labeled partition 1, even though it's combined with 2 in this partition assignment. And you've got another partition labeled 2, all right? So for the two partitions together, you calculate or estimate the shortest tree for random partition 1. So for all of the characters labeled 1 in this data set, you take those out and you estimate the tree based on that partition. And let's say that this random partition for only the characters labeled 1 from partition 1 has a length of 39. And let's say that your character partition 2, the length for the tree from that particular partition is, let's say, 46. This gives you a length of the combined partitions, once they've been combined, of 85 steps. Okay, we can chat more about this when we meet face to face. So what the ILD test does is it repeats this between 100 and 1,000 times. It combines randomly those characters or characters from data set 1 or partition 1 and 2, and then it looks at the trees when you've combined these data sets. So what's going to happen then is you are going to get a figure that looks very much like this. This is what the program spits out at you, and you can kind of look at it a little bit more closely. On the y-axis, we've got number of partitions. So let's assume that this program, the ILD program, ran 1,000 different partitions, where it built 1,000 different pseudo-replicate partition data sets. And we know that the original data, the original sum of the two trees separate was 68. So the original data were the two trees when analyzed separately. Okay, so the two trees analyzed separately, their sum was 68. What this graph is showing you is that for the 1,000 number of random partitions that were created, we have trees that range anywhere between 79 and 87 steps long. The first tree example that I pointed out was we found a tree that was 85 steps long, but we also luckily found some that were 79 steps long. What is this suggesting to you? After running 1,000 different pseudo replicates or pseudo partitions, when we combine partition 1 and 2, is that in no instance did we find any trees that were shorter than 68. And what that means is that when we combine the data, the two partitions, into a pseudo combined data set, we never retrieve a tree that is more parsimonious than when they were run separately. What this is suggesting is that no trees exist that are more parsimonious than when the data sets or the partitions were run or analyzed separately. What this suggests, guys, is that you should not combine these data sets, because all they do, based on these random 1,000 number of pseudo partitions, is you get longer trees. So this suggests that you should keep the data or keep the two partitions separate. Okay, and just as a little terminological note, when you hear the term concatenate, that refers to the combination of data. Basically, the ILD test in this particular example is telling us that we should not be concatenating the partitions. We should run them separately. So let's go back to the carnivore data. All right, we talked about carnivores as a whole, and we know that the molecules and the morphology don't match up. We did another test, the Templeton test, that helped us decide that the molecular tree is actually quite suitable, even for the morphological data. So what if we ran the same analysis with our carnivore example? Here is the output of that particular study. All right, so imagine that 1,000 replicates were run in which the morphological and the molecular partitions were combined. So these are the results. This is from the morphological and the molecular data combined. In other words, the two partitions have been concatenated and run together. Okay? So out of all of the different partitions, imagine that this is the output that we get. And I didn't tell you this earlier, before the partitions were combined, when you took the morphology and the molecular tree and you put them together, they gave us a sum of 820 steps. So this was when the two partitions were run separately, but when you sum those two tree lengths together, they give you 820. So let's say the morphology tree was 30 steps and the molecular tree was, say, 790 steps. That would give us 820. You will notice that something interesting happened. When these data were combined, look at this. We actually got a couple of trees that were shorter than the original separately run partitions. When the partitions were concatenated and run together, we ended up with a tree that is actually better. A couple of trees, as a matter of fact. And then there's a whole bunch that are way longer, okay? So if we think of this as we ran 1,000 different partitions random partitions in the ILD test. And let's imagine that there are 8 in this range of 820 steps plus the couple that are in the 819 step range. There were a total of 85 trees that are equally good, that are equally parsimonious or shorter than when the data were run separate, than when the partitions were analyzed separately. Okay, so 85 out of 1,000 different trees that we looked at, 85 of those gave us a tree that was just as good or shorter than the original set of trees. And if you do the math here, 85 divided by 1,000, you will end up with the equivalent of a p-value of 0.085. All right, or 8.5%. Think about whether this is significant or not. Basically, what we can assume here, just based on the figure, is that you can combine the data and get shorter trees, or trees that are just as good as when you run them separately, or that the two data sets have about an 8.5% chance of having conflict. All right, so essentially what this is showing you guys is that you should combine these data sets and you could get a more parsimonious tree. And you can do the same for maximum likelihood. As long as you can interpret a figure like this, or if I gave you a set of partitions and you had to discern whether you should combine or not, you should be in good shape. So please make sure that you understand the difference between the ILD test and the Templeton topology test. They are looking at different things. One, the Templeton test is helping you decide whether the two data sets are significantly different or not, and whether one data set might actually fit the tree of another data set or a partition. Whereas the ILD test helps you to discover whether you should perhaps combine the data and get a tree that might be a little bit better. So at the end of all of this, all I want you to understand is that in the realm of all the different tree possibilities, and in the realm of all of the complex things that we know about DNA, morphology, and potentially any other types of data, we could get different trees that give us different answers. And it's always good to either combine the data and look at more. The more data that you have could sometimes pose problems because the analyses take a long time. But if those data together are giving you a good signal about the phylogeny or the potential phylogenetic patterns that exist in those organisms, that is a great solution to these issues. And so, final lesson, use as much data as you can. Do these tests to discover whether you should combine your data, and never reject a data set just because it doesn't give you the most parsimonious tree. Okay, so please come with questions. I look forward to seeing you all on Wednesday, and good luck with everything. By the way, if you got annoyed by my scratching, I have a new drawing tablet that I've been using. I hope to get back to the Surface Pro soon. Otherwise, I will see you all later, and I miss you. Bye.